Aluminum toxic effects and how aluminum impairs methylation. We're now focusing on M6A RNA methylation. So M6 RNA is a major internal modifier of messenger RNA. It has a critical role in RNA metabolism, such as how they are translated, transported, degraded, or stabilized in methyltransferases incorporate M6A. So this study shows that aluminum exposure was associated with M6A RNA modification in mice with significant neuronal injuries of the hippocampus, reduction in the M6A RNA modification, and expression of methylase. Okay, so M6A RNA modification had a crucial role in developmental neurotoxicity induced by aluminum. I want to talk about some important things that the study notes and background information, and then we'll dive into the study and the results in the next post. Okay, so they note that there is evidence that suggests that aluminum exposure during pregnancy and lactation can affect the development of fetuses in the uterus and postpartum in infants. So the damage of neurons during this critical period may be long-lasting, and aluminum exposure during this time has been potentially harmful. It says that aluminum accumulates in the brain and cerebellum in an age-related manner. It can cross the blood-brain barrier and accumulate in the fetal brain tissue, where it remains throughout the lifetime of the organism. They looked at the effects of aluminum on hippocampal neurons of mice offspring. They gave pregnant mice aluminum lactate in their drinking water from gestational days 6 to postnatal days 21, and then the offspring were examined postnatal days 21 and 60. Picking up with a study looking at the effects aluminum had on mice when exposed during pregnancy. One of the things they looked at were Nissl bodies, and these are structures and neurons that are essential to proteins, and these were reduced in the aluminum exposure group postnatal days 21 and 60. Okay, there was a decline in the density of dendritic spines in the aluminum treatment group, indicating that aluminum exposure led to irreversible neuronal damage in the developmental stages of mice. They also looked at DNA fragmentation to indicate apoptosis or cell death, and that was increased in the aluminum group by 1.2-fold, indicating that aluminum exposure during developmental stages increases apoptosis. They determined the levels of M6A RNA methylation, which were decreased in the aluminum exposure group. You can see those findings in these figures here of control versus aluminum. And then they also looked at embryonic exposure to adult stages. So mice that were exposed to aluminum day 6 gestation, then to offspring at day 60. Okay, that showed that Nissl bodies of the hippocampus decreased by 30% in the aluminum exposure group. There was a decrease in density of dendritic spines, and the level of M6A RNA methylation was decreased by 64% in the aluminum exposure group. There was also decreases in RNA expression of levels of METTL3, METT14, and YTHDF2, suggestive that M6A RNA is involved in neurotoxicity induced by aluminum. So this is a very well laid out study, and one of the more upsetting findings I came across, because this study is showing that aluminum exposure during pregnancy induces developmental toxicity and could persist into adulthood despite no substantial aluminum accumulation. So basically, it doesn't take cumulative exposure, just one during pregnancy. We're continuing to look at how aluminum impacts methylation with focus on RNA methylation. This builds off of how aluminum affects M6A methylation specifically. So the study demonstrated that M6A methylation decreased with aluminum exposure. For a refresher, M6A is that internal modification of messenger RNA, and there are different proteins that add and remove M6A, such as METTL3. They gave mice aluminum for 12 weeks and then looked at the hippocampus. So the figures here show the accumulation of aluminum. Neurons show deformation. Nissl bodies were reduced by 18%. There was also an increase in apoptosis or cell death. This is seen in figure D, and also found that it was in a dose-dependent manner. So there was a 1.5, 2.7, and 3.2-fold increase in cell death with higher aluminum concentrations, as you see in figure B with the increased aluminum concentrations from left to right, demonstrating that METTL3 mediated M6A methylation. It's a critical regulator in the development of neurotoxicity induced by aluminum exposure, and insight into how aluminum exposure impacts epigenetic regulation of neurotoxicity, or how those genes are affected or changed by being exposed to aluminum. Methylation is happening everywhere in our bodies. So throughout the section of impaired methylation in my graphic, many of the studies looked at methylation in the brain, because methylation in the brain can affect the entire body with downstream effects, so in an indirect way. But methylation also directly affects everywhere because it's continuously happening in every cell. So this study is an example of how aluminum impacts methylation that affects asthma. The study showed a significant association between high blood aluminum and high ADRB2 receptor methylation, which is a receptor involved in the pathogenesis of asthma. And higher aluminum concentration was independently associated with an increased risk of uncontrolled bronchial asthma. And bronchial asthma is known to be mediated by epigenetics. ADRB2 receptor is in respiratory tissues and the target of bronchodilators by being a beta agonist. So that's what causes relaxation of the airways. They measured methylation and aluminum concentration in the blood of 70 kids. You can see here in figure 2 that the asthmatic children had significantly higher aluminum and methylation that also corresponded with lung function by looking at spirometry. And those with higher aluminum levels had worse asthma control. So I wanted to share this as an example to show that methylation changes aren't just in the brain, but throughout the entire body with various effects.